Hello, everyone. Thanks a lot for the super nice presentation, Martin. As he was saying, my name is Laura, and it is my pleasure to be here to be able to present my master thesis project. So the title of this project is Microvascularized Neurovascular Unit Model using human-induced pluripotent stem cells and laser cavitation molding, which may sound a bit too buoyant or too much, but let me cut it down for you. In this presentation, I will be mainly focusing on why and how did we use human-induced pluripotent stem cells, and how did we leverage the use of laser cavitation molding. I'm not sure you're seeing the squares right. I am not, but anyway, in any case. But before we go into that, I would like to talk to you about the model itself. So for those of you who are not familiar with brain anatomy, this is the neurovascular unit of the brain. It is composed of very different cell types, like neurons and astrocytes, but also endothelial cells, and they're arranged in a very complex and organized way. So for example, you can see here the endothelial cells are surrounded by the support cells, which are pericytes, which in turn are in close contact with other brain cells around them. This unit is actually affected by many neurological diseases like epilepsy or stroke, which are actually a major cause of death and disability worldwide. But yet there's a very big knowledge gap regarding these diseases, and it's due to the lack of suitable brain in vitro models. So if we want to know more about these diseases and the brain physiology in general, we need to develop more relevant and biomimetic models of the brain. However, some challenges you may encounter when developing a neurovascular unit of the brain are, first of all, the cell source. As you can imagine, obtaining brain cells for research is not easy due to the invasiveness of the technique. So working with a human cell source is not trivial. And also another issue is the vascularization, which is actually a common challenge in many other in vitro models, but in this specific case, it's even more relevant due to the key role that the vasculature plays in the neurovascular unit. So this project is mainly focused on tackling these two challenges. On the one side, we have the cell source. In this case, in this project, what we used were hiPSCs, or human-induced pluripotent stem cells, to generate brain organoids on one side and vascular cells on the other side. Now, the good thing about this is that all cellular components belong to the same patient, and so that allows us to generate a personalized in vitro platform of this person's neurovascular unit. Now that we have all the cellular components in place, we want to put them in a chip. So in this case, we use the commercially available chip developed by AIM Biotech, the Identics, which is composed by a central compartment flanked by two media channels. So the idea is to get the brain organoids, embed them in a hydrogel solution, and cast this solution in the central compartment of the chip. Now that we have the brain organoids inside of our chip, we want to vascularize this model. And this is where the laser technology comes in place. So the idea is to pattern some channels around the organoids that will connect both media channels in the chip. And of course, in a later stage, as final step, we want to seed the vascular cells inside of these channels, right? So now that you have a bit of a good overview of what the main goal of this project is, let's move on to how do we make it happen. So let me start first with the cell culture part. To produce vascular cells, we use human-induced pluripotent stem cells, and we made use of the differentiation protocol developed by Pars and her group. So the whole thing starts with a skin biopsy. The skin cells are reprogrammed back to a pluripotent state in the IPS core facility at Karolinska Institute. And so we get these reprogrammed cells in the lab. We make them go through a mesoderm-like state, and later on through a vascular specification, rendering a mixed population of vascular cells. Now we want to have a population of endothelial cells separated from the rest of the cells, which are support cells, and what we do to obtain that is this MAX, or magnetic activated cell sorting, using CD31 as a discriminating protein. So the whole process takes up about 12 days, and we end up with a population of CD31 positive cells on one side and a population of CD31 negative cells on the other side. So this is how the theory looks like, but of course when you go into the lab, things don't always work the way you would expect. So what we need to do before anything else is to assess the identity and functionality of these cells. And I don't know how familiar you are with cell culture, but performing this characterization and generating this data took up most of the time of this project. And because just simply stem cell culture is just, it takes insane amount of work and time, so much so that these results that I'm showing you here would not have been made possible without the help of my colleague Monica, that you can meet in that picture. So yeah, as I was saying, we will assess the identity of these cells by performing some immunofluorescence stainings on them. And let me tell you a bit about the strategy that we followed. So we have a plate of CD31 positive cells on one side and another plate of CD31 negative cells. We stained each cell type with 2 specific markers, expecting some positive signals, but we also stained each cell type with a gold standard marker of the opposite cell type as a negative control. So you can see the results here. On the top row, you have our population of CD31 positive cells, which are positive for CD31 and VCAT, but are negative for PTGFR, which is a good sign already. And on the bottom row, we have our population of CD31 negative cells, which are negative for CD31 and positive for PTGFR, which is a specific marker of pericytes, but they're not positive for alpha smooth muscle, which is contrary to what we were expecting, but this only means that we're not rendering a pure population of pericytes, but rather a more mixed population of cells. So we definitely will be looking into this further. Apart from these immunofluorescence stainings, we also performed some functional assays, like the 2D tube formation, which is an assay that is usually performed on endothelial cells to test their ability to form networks. The picture that you're seeing are differentiated endothelial cells forming these networks that I was telling you about. So also a good confirmation. Now, yeah, that was it for the biology part. Let me tell you a bit more now about the other very important part of this project, which is the femtosecond laser. So in the schematic, you're seeing the design that we followed when patterning our channels in the chip. And then close to it, you're seeing a video of what it really looks like in real life. So the phenomenon that you're seeing in that video is called laser-based cavitation molding. And this happens when a very fast and high energy pulse of light is irradiated on a hydrogel. As you may know, hydrogels are mainly composed of water. So when these hydrogels are irradiated with this pulse of light, what happens is that water is evaporated really fast in a very local region of the hydrogel, and that generates a bubble that expands. So as the laser moves through the hydrogel, new bubbles are formed, and they restructure. What they do is that they restructure the matrix of the hydrogel, leaving an open channel behind. Now, the attractive part of this technology is that it's also a cell-friendly technology because it doesn't ablate the hydrogel, but it rather rearranges or restructures the polymers in the matrix. Proof of this was performed by my supervisor, by Alessandro, in which he put some cancer cells in the hydrogels, cast the hydrogels in the chips, and then patterned some channels of different sizes, like 20 micrometers, 40, and 60 micrometers in diameter. And, well, he then ran some viability assays, like you can see here in the graph. And what he proved, or what he saw, is that viability remains really high in regions in close proximity to the pattern channels, as close as 20 micrometers, with viability rates of 80% in any case. Another biological demonstrator of the biocompatibility of this technology was this experiment. In this case, he put some glioblastoma spheroids inside a collagen hydrogel, casted this collagen hydrogel inside of the chips, and then again patterned some channels around it. And then what he did was to seed HUVECs inside of the channels and let them grow in for 3 days. So in this picture, we can clearly see how the HUVECs are sitting inside of the channels and covering them in as short as 3 days, but also the cells from the spheroids are invading the matrix and moving towards the channels. So as I have already told you, most of my time in this project has been devoted to optimize the differentiation protocol with the cells and implement the laser technology for this specific application, but I still had some time to combine them both, and these are some very preliminary results that I obtained. So in this case, what I did was to prepare some 4 milligrams per ml collagen hydrogels, cast them inside of the AIM Biotech chip, and then pattern these hydrogels. So I obtained these very thin and continuous channels that you can see in the image. And later on, I seeded a population of CD31 positive cells in some of the chips, and I mixed CD31 plus and minus cells, and I seeded them on another group of chips. So what we can see here is that these are days 1 and 3, as you can see as well. And what we can see is that for the monoculture of cells, cells are actually covering the channels that I patterned and trying to invade and migrate outside of the channels towards the matrix, which is actually a really good sign from the point of view of the functionality of these cells, but it's also a good sign for the application, the specific application of this project, in which we want our future organoids to be vascularized by the surrounding channels. In the case of the co-culture, we're seeing a much more heterogeneous population of cells, but they also show a tendency to migrate outside of the channels, although in a different trend than what CD31 cells do. But again, these are very premature results, and we need to perform more experiments and look further into this. So with that, I would like to sum up this presentation and go through the most important milestones that we achieved in this project. On the one hand, we optimized the differentiation protocol to obtain vascular cells from iPSCs in a consistent way for different donors, obtaining a pure population of endothelial-like cells, which proves the robustness of the protocol. And on the other side, we obtained some very promising but early results when seeding our cells, our differentiated cells inside of the channels, as we saw how they tended to migrate inside of the collagen and outside of the channels. And even if these are no minor achievements, there's still a lot of work to do. So right now we're currently focused on further characterizing the differentiated cells in a more exhaustive way, but future plans include the incorporation of the brain organoids in the chip, the optimization of a suitable media for co-cultures, because at some point we will have very different cell types in our chips. And also we're exploring other microfluidic platform designs, like the chip that my supervisor Julia developed, that you can see there. And of course, in the long run, it is our intention to develop a disease model of GLUT1 deficiency syndrome. But this topic may be a bit out of the scope of this talk, so if you really want to know more about this and about this disease and this super cool chip, you should definitely go by poster 439 tomorrow. Julia will be presenting for you there. And with that, I would like to conclude my talk and give a special thanks to my supervisors, Anna and Frank, for giving me this opportunity to be here, for Julia and Alessandro for the constant support and trust, and of course to my colleagues Monica and Raluca. Also special mention to the Nanobiotechnology Division and the Micro and Nanosystems Department in KDH and all the people working there. And of course to you all for your time and attention. Just if you would like to know more about this project in particular, you should definitely pass by poster 439 again. And regarding the laser cavitation molding technology, here's a QR code for Alessandro's paper, in case you want to know more details on how the technology works. So yeah, thanks again, and the floor is open to any questions.
Okay, thank you, Laura. Thanks to you, we saved 2 minutes. So we are on time again. Questions, I see one already here.
Thank you for your presentation. What I wanted to ask about is the advantage of your fabrication method. For example, I have seen some publications about the vascularization of particularly brain organoids. So what advantage would your system bring, I wonder?
Yeah, thanks for the question. I would say mainly it's the viability of the cells after patterning, because I know some other techniques using laser technologies are more aggressive to the cells. So I would say that for sure. It's also a fast technology when it comes to pattern channels in these type of vessels. And also, if I have to think about it now, yeah, I can think of those 2 advantages for sure. And also you can generate channels in a reliable and on the long run, let's say, way, like they stay stable for a long period of time as well, which is something that is not trivial.
How long have you cultured the cells for? Maybe I didn't see it.
Yeah, I think the longest, the only experiments I performed, it's been the seeding inside of the pattern channels, and I think the longest it's been for 10 to 12 days, but I still need to run some viability assays on the cells as well. Of course, I just limit it to observation. Yeah.
Okay, we have another question here.
Hey, congrats on your impressive work. I had a question regarding the cell source that you use for your project. You said different cell lines. Can you comment on how many cell lines, whether they were gender specific, and whether they were from skin or from blood-derived iPS?
Yeah. Yeah. The cell, well, I don't like to call them cell lines, let's say, for to not confuse the terms, but yes, we use different cell sources. And so as our intention is to progressively move towards a disease model, or to use this platform for a disease model, we're working in parallel with patient cell lines or cell sources and donor cell sources. And currently, most of them are males, but we're working with a female, and I think some of them are also kids or young people of a young age, but also adults as well. So we are also paying attention to the variability in that sense. Yeah.
Okay, one last question for Laura.
Thanks a lot for your talk. It was very interesting. And since you said that your technology takes advantage of hydrogel's property to retain water, how does the yield or outcome depend on the hydrogel type and its formulation?
Yeah, that's a good question. We're also looking into that because so far only work has been done on collagen type 1. But we're looking into other collagen or other hydrogel formulations like hyaluronic acid and fibrin gels as well, and different concentrations of collagen for sure. We've seen promising results for collagen hydrogels of different concentrations, but of course, definitely it affects the polymer concentration in the hydrogel for the outcome. Yeah.
Okay, let's thank Laura again, and we move on to our next speaker.